If we're going to understand biomolecular mechanisms, we need to have a good handle on the sorts of bonds and interactions it can form. And so the goal of this video is to dig into that. So let's get started. So we're going to take all of the interactions that biomolecules can form, and we're going to sort of break them into a tree that helps us organize them and think about them. So at the broadest level, we can see a differentiation between covalent and non-covalent interactions. So covalent bonds are ones where electrons are shared between atoms. So we've got here a double bond. We've got electrons that are being shared across these orbitals, whereas non-covalent interactions do not involve electrons being shared. In this case, we have a positively charged amine that's electrostatically or electrically, coulombically interacting with the negatively charged carboxylate. They differ in their energy, so the energy to break a covalent bond can be from 3,000 to 1,000 kilojoules per mole, so quite a bit of energy to break one of these bonds, whereas non-covalent bonds, maybe it's only 1 to 50 kilojoules per mole to break, so much less energetically costly to break a non-covalent bond than a covalent bond. And then they also have different barrier heights. So the barrier height to break a non-covalent bond is high, meaning there's a big energy barrier it has to go over. It is energetically expensive and therefore hard to do. It's a slow reaction to break that bond. Whereas in non-covalent interactions, they tend to have low energy barriers, so they're easy, they're fast to break. This leads to a really good biochemical rule of thumb when you're looking at some complicated biochemical pathway or reaction or mechanism. If you see a covalent bond that's being formed or broken, it almost certainly requires an enzyme to do so, whereas if you see non-covalent bonds being formed and broken, it probably does not require an enzyme to do so. So it's a good sort of strong break across biochemical mechanisms. Now, if you've taken organic chemistry recently, you've been obsessed with covalent bonds, right? There's so much of that typical coursework involves breaking bonds and forming bonds and cool reactions. Biology, by contrast, really loves non-covalent interactions. So this is a painting of actually a viral infection, so maybe not that happy, but it's a painting by David Goodsall, and we have all of these thousands of proteins and small molecules and nucleic acids and lipids, and they're all bouncing into each other, interacting transiently, and this allows the fact that all these interactions are non-covalent allows the cell to respond to its environment. It's not like one fixed rigid thing that once it happens, it happens. No, proteins can form a complex and then they can break apart. DNA can wrap around a nucleosome, it can come off. A bilayer can be fluid because the molecules can move relative to each other. So if we're going to understand biochemistry, we really have to dig deep into understanding non-covalent interactions. So basically, we've talked about covalent bonds over here. They're strong, they're slow to break, they require enzymes to break. And now we're going to spend the entire rest of this video talking about non-covalent interactions because it turns out they're critically important for understanding biology. If we take non-covalent interactions and break them in half, we would say there's two broad categories. There's polar interactions and there are non-polar interactions. And what I'm going to do is actually unpack them individually. So first, we're going to talk about hydrogen bonds as the example par excellence of a polar interaction. And also probably, if I had to bet, I would say the most important polar interactions in biochemistry. So hydrogen bonds, often abbreviated as H bonds, they form between the lone pairs of electronegative atoms like oxygen and hydrogens that are bound to nitrogen or oxygen. If we look at this carbonyl down here, we have an electronegative oxygen bound to a less electronegative carbon. What that means is electrons will spend more time with the oxygen than they will with the carbon, creating a partial negative charge on the oxygen. We then bring an amine over here. This amine has the same kind of phenomena happening where you have an electronegative nitrogen that is sucking electrons off of this proton, making it partially positive. These can then form a hydrogen bond interaction. By convention, at least in all of my content, but across much of biochemistry, we draw hydrogen bonds as yellow dashed lines. So that's how you're going to see it in all of my content. But this is a relatively favorable interaction. It's polar because it involves bonds that are polar, right? You've got a negative side interacting with a positive charge. Positive side, you can tell which way the bond is facing. We use some words to denote which way the bond is facing. Anytime you have something that's binding to a proton in a hydrogen bond, that will be the hydrogen bond acceptor, whereas the side that's donating the proton will be known as a donor. So we have a donor-acceptor pair that together form a hydrogen bond. If you look across biomolecules, this covers most of the acceptors. You have these classes of molecules. Basically, anytime you see an oxygen or a nitrogen with a lone pair, that lone pair can act as an acceptor. And then most proton donors, hydrogen bond donors in biochemistry, are attached to either a nitrogen or an oxygen. So anytime you see a hydrogen dangling off of an electronegative atom, an oxygen or nitrogen, you can say, oh, that could be a hydrogen bond donor. One of the things that's really important to keep in mind, and the reason that hydrogen bonds have this outsized importance, is that there is a 55 molar hydrogen bonder floating around in water, right? And water can be both a donor and acceptor. And so this plays a huge role in shaping how biochemistry works, because all the time, every hydrogen bond is having to worry about, am I bonding to a protein, or am I bonding to water, or am I bonding to DNA? But water is this like everywhere all the time sort of thing we're worrying about. So how strong is a hydrogen bond? And it turns out it depends on things like the distance and the angle and the environment. But I think a really good rule of thumb is that a water-water hydrogen bond is about 20 kilojoules per mole, which means at a physiological temperature, it's a human physiological temperature of 310 Kelvin, that means about 1 out of every 2,000 to 400 hydrogen bonds are broken. So of the bonds that are possible to form, 2,400 of them are formed, and then occasionally one is broken. So it's a relatively strong individual bond, but it's not outlandish, right? The odds of a 1 in a 2,000 event aren't crazy when you're talking about something where there's thousands of molecules involved. Okay, that was the hydrogen bond. What about the example par excellence of a nonpolar interaction? It turns out, I would say that's the hydrophobic effect. So let's unpack that. To unpack the hydrophobic effect, let's first imagine we have taken a benzene and we put it randomly into a box of water. In fact, that's what I did. This is a molecular simulation where we've got a bunch of benzene floating around, a bunch of water molecules. The benzenes are shown as gray circles with hydrogens hanging off of them. The waters are these triangle things of oxygens and hydrogens. Now what I'm going to do is let the simulation run. And what I'd like you to think about is what's going to happen? What's going to happen and why? So give you a chance to pause the video. Okay, I'm assuming you paused it. What we see is the benzenes all clumped together. They're clumping together to the exclusion of water. So we have this whole domain here that's basically just benzenes. And then there's this surface where some of the benzenes are interacting with water. Why? What is happening that's causing this? And it turns out that what's happening is mediated by the water more than it's mediated by the benzene. And so a hydrophobic molecule like benzene disrupts hydrogen bond networks inside of water. So here I'm showing a simulation with a single benzene sitting in a box of water. And what I'm going to do is remove a bunch of the waters and just highlight some of the hydrogen bonding patterns. And what we can see here is the benzene has its direct neighbor. It's touching this water here with these circles this water I've drawn with these small spheres. And you'll notice that water has 2 hydrogen bond partners. Whereas this water that's distant from the benzene is able to form 4 hydrogen bond partners. And in fact, this holds true statistically. If we just calculate over, say, a molecular simulation, what's the distance between water and benzene, and how many hydrogen bonds does it have, what we see is there's a pronounced dip right in the shell, right in the region right around the benzene, where we have fewer water molecules fewer hydrogen bonds per water molecule as opposed to out in the bulk. So the hydrophobic molecule is disrupting the hydrogen bond network around waters. Well, then what does that mean? Well, what it means is that if you can clump together hydrophobic molecules, you can release water molecules, right? Just as a geometry problem, if I look at, say, every one of these benzenes in the side on the left has a bunch of waters near it. If you count on average, there's 13.7 waters next to every benzene. Whereas when you start clumping benzene molecules together, this one in the middle doesn't have to interact with any waters at all. And what that means is, at least at this point in this simulation, that we've only got 10.3 waters per benzene. So we see less of a disruption of those hydrogen bond networks when we clump the hydrophobic molecules together. So what we can think about is the hydrophobic effect is the tendency of hydrophobic molecules to come together and release water. So I've got, drawing this schematically here, I've got a hydrophobic molecule that's bound by 4 waters, another hydrophobic molecule, the molecule that's bound by 4 waters. If I can jam those together, 2 of the waters get released and can interact with themselves and are no longer part of this disrupted network. And so that's an energetically favorable process. How strong is the hydrophobic effect? Well, this depends on a lot of factors, but it depends on things like the surface geometry, things like temperature and pressure. There's all kinds of complexities. But A good rule of thumb that people use in things like drug design is at physiological temperatures, you get about 0.15 kilojoule per mole per angstrom squared of exposed surface area. So if I expose hydrophobic surface, it's going to cost that much per angstrom squared, which roughly works out. If you bury a carbon atom, it's about 10 kilojoules per mole. And that's a very, very rough number. We should put, all kinds of caveats on that. But it's close. It's within an order of magnitude. And one of the interesting things about that is the hydrophobic effect order of magnitude. I don't know if you remember, the hydrogen bond was about 20 kilojoules per mole. The hydrophobic effect is about 10 kilojoules per mole per buried carbon. So we have these two competing effects, or these two different effects that have very similar magnitudes, but very different molecular bases. So hydrogen bonds are between different biomolecules with positive and negative charges, or partial positives and negatives. And these are between non-polar molecules inside of a aqueous environment. Okay, you might be looking at the time in this video saying, oh man, this is going to go on forever. Don't worry, we're just going to now just zip through the remaining interactions. So the hydrogen bonding and the hydrophobic effect are responsible for the lion's share of the interactions that we have to worry about inside of biochemistry. There are other molecules, other interactions that are really important and become very important if you say want to build a drug to bind to a specific target, but these tend to be specific cases, right? If you want to understand the general features of a system, you want to understand hydrogen bonds and hydrophobic effect. If you want to understand the details, you have to start adding these other components. So really quickly, what are those other components? We have ion pairs, which would just be, we actually already saw one, a formally positively charged amine interacting with a formally negatively charged carboxylate. That's an ion pair. A cation pi interaction is a fun one. It's where you have, say, a positively charged amine that nestles into the electron clouds of an aromatic residue. So there's a pi orbitals with electrons. They can interact in a favorable way. On the nonpolar side, we have van der Waals, which is the tendency of atoms to want to pack together, but not overlap, right? You can bring molecules, you can think about them as hard spheres that want to knock into each other, but as soon as you try to take those molecules and push them into each other, they repel. So that's van der Waals interactions. And then you also see things like pi-pi stacks, which would be two hydrophobic aromatic ring structures that pack on top of each other. They share electron, or they can polarize in response to each other and cause a favorable interaction. But again, these are important details. These are the key to understanding 90% of the biochemical sort of interactions and interact mechanisms. So in general, that's where we're going to focus. Last thing I want to point out, and then we'll be done, is that nonpolar and polar interactions actually play different biological roles. This idea is so important, actually, I'm going to have a whole video on it. But just to hint at it, it's that nonpolar interactions tend to make molecules stick together. In an aqueous environment where there's a lot of water around, if you have hydrophobic surfaces, they're just going to want to stick, right? And they're going to just stick randomly. Any hydrophobic surfaces will stick to any other hydrophobic surface. Whereas hydrogen bonds and other polar interactions make sure that you've got complementarity, right? You have to have a positive interacting with a negative, a donor interacting with an acceptor. You can't just have them willy-nilly sticking together. So the hydrophobic effect tends to drive sticking, and polar interactions tend to give specificity to what sticks to each other. Okay, so key points from this. Biology is built on non-covalent interactions. The most important ones, I would say, are hydrogen bonds, which is when a proton donor finds lone pair acceptors. On the nonpolar side, we have the hydrophobic effect, where hydrophobic surfaces interact with each other, releasing water. That's a very favorable process. And finally, the nonpolar and polar interactions have different biological roles, driving either sticking or sticking specificity. Okay, well, hopefully you enjoyed that quick tour of biomolecular interactions. We're going to unpack these in future videos, for example, looking at how receptors recognize specific ligands using these interactions. And again, we're just going to encounter this as we're going through all of biochemistry. Like, oh, look, there's a hydrogen bond. Hopefully now you have a better picture of what that is. So thanks. I will see you later.